Hi, we are Karim, Alfredo, and Laura, and for this presentation, we are going to talk about the reduction of particle trapping voltage needed in an insulator-based dielectrophoresis. We will start with the introduction of the technique, the objectives of the project, and the theoretical background. Then it will be presented the methodology and the first model as a precedent of the model, and finally the results, discussion, and conclusions.
Well, I'm gonna start explaining the introduction, commencing with explaining what a particle trap is. The particle trap is a device that is used to confine charge or neutral particles, and their interaction with the wall of a channel or a container has to be avoided at all costs. One of the main purposes of these devices is to study the properties or possible interactions of the particles at stake within certain environments, and also to characterize them. Some particle trapping methods exploit the particle properties such as deformability, magnetic susceptibility, density, size, polarizability, and also shape. This is done by applying the corresponding force fields such as electric, magnetic, acoustic, optic, and hydrodynamic. So next I'm going to explain the insulator-based dielectrophoresis. This is a microfluidic technique that employs the uses of electrodes and or insulators to create the dielectrophoretic forces. The technique is used mainly for particle analysis and has a wide array of applications. Some of these are the separation of bioparticles like bacteria from mixtures, detection of mycosis, and also separation of cancerous cells by their physical factors or properties. The basic structure of an iDEP consists of a microchannel in which the particle containing fluid will flow. It also contains three-dimensional structures which will act as insulators and thus generate the non-uniform electric fields. These fields will trap the particles of interest. Attached to the ends of the devices are the electrodes. Examples of some microfluidic particle trapping and sorting methods are magnetophoresis, acoustophoresis, optical tweezers, hydrodynamic filtration, and hydrophoretic focusing. But as our goal is to reduce the voltage needed in order to trap the particles within a microfluidic channel using electric fields and not acoustic or magnetic ones, we as a team decided to use the method of dielectrophoresis. This one involves the manipulation of electric fields for the particle isolation. Some recent contributions using insulator-based dielectrophoretic system have been delivering promising results, such as models that allow for trapping, switching, and storing particles all in the same device. Others for the identification and collection of pathogens, such as Listeria, and others for characterization of single cells and particles.
As mentioned before, the main objective of this experiment is to improve the results of the paper Simple Approach to Reducing Particle Trapping Voltage in Insulator-Based Dielectrophoretic Systems by reducing 30% of the voltage needed in the insulator-based dielectrophoretic of that experiment. The development of microfluidics started at 1980s when there was its first application by the generalization of the inkjet printing, and currently most biotechnological developments are based on microfluidics, basically because biological samples are in a liquid state and biological organisms live in an aqueous environment. An important challenge for this development is the complexity of the microfabrication to correctly get a volume reduction from the macroscopic environment and concentrate it in order to be introduced in biochips, also considering the most useful material. For the theoretical background, it is important to talk about the electrokinetic phenomena present in microsystems. This phenomena is studied by electrohydrodynamics, which is the coupling between an electric field and the movement of particles or fluids. Electrophoresis is defined as the movement of charged particles by an electric field distributed in a solution. This displacement is made with a mobility that depends on the characteristics of the charged object. The dielectrophoresis is the lateral motion impart on uncharged and polarizable particles once the polarization is induced by non-uniform electric fields and of other uncharged and polarizable particles as a result from the interaction between those electric fields. Electroosmosis appears in the presence of established double layer when an electric field is applied parallel to the surfaces. The double layer is moved by the effect of Coulomb forces, and the electrokinetic flow around stationary surfaces is known as electroosmotic flow. Electric current measures the displacement of the charge, which can be displaced by diffusion governed by Fick's law, which compound with conservation of mass equation and permittivity give us the equation governing the flow of a charged fluid, known as the Navier-Stokes equation, shown in equation 1. It is important to notice that the fluid is subjected to an electric field. Well, in order to get the replication and improvement of the model, it was used the software Thomson Multiphysics 5.5 for the creation of a two-dimensional model with two channels by using a channel length of 10.16 millimeters and width of 8,050 micrometers. One channel with one column and the other one with three columns, both with 6 insulating posts each. The elliptically shaped insulating posts have a minor axis X of 35 micrometers and a major axis of 65.8 micrometers, with a spacing between the columns of 37.5 micrometers and 12.08 micrometers. The materials used was PDMS and water as the fluid, and the inlet voltage was reduced to 690 volts.
As a base model for our improvement, we use the one programmed in the paper Simple Approach to Reducing Particle Trapping Voltage in Insulator-Based Dielectrophoretic Systems. In this paper, as a system was developed with different arrays of columns, consisting each column of three insulating posts at the center and two halves, each one at each end of the device. As results, they obtained the following graphs displayed here, A and B. In A, we can see the electric potential and how the stiffness changes depending on the column contained in the array. It is worth mentioning that the array with the steepest slope was the one with one column of posts. In figure B, we can see the electric field intensity. We can see that the intensity of the array with one column was the highest, and the one with 21 columns was the lowest.
We tried to replicate and improve a previous model which obtained the following results. For a chamber with a single column, they used a voltage inlet of 1000 volts. In the electric potential chart, we notice there's an electric potential drop at around 5000 micrometers in the x-coordinate, which represents a depolarization of the system. On the other hand, the electrical field increases to 6.5 volts per meter at the constriction region. This was our minimum goal. So, after changing the geometry of the system, these are the results we obtained. With 31% less of input voltage, 690 volts to be exact, we managed to enable the device to operate with even smaller voltage while increasing the electric field intensity constant in the constriction region. As you can see in the figure to your right, the electric field increased from 6.5 to almost 8.5 volts per meter. This phenomena occurs because of the close gap between the posts. As closer they get, the stronger the electric field becomes. However, the gap cannot be too narrow because then the fluid won't flow and the electric potential will get bigger. Now, as for the 3-column array, we see that with an input of 1000 volts and a significantly electric potential drop, the electric field reach was around 6 volts per meter. However, with our changes, next slide please, we reach a peak of electric field intensity at 9 volts per meter with 310 volts less of voltage input. So, in conclusion, the electric field intensity was successfully increased in the construction region, while inlet voltage was reduced up to 31% by adding 2 insulating posts to the columns and reducing the gap between them. Thank you very much.